Hi everyone. Sorry about that. If you're just tuning in, what I don't know what just happened, but the stream that we were just doing got cut off. Not sure how that happened. It's something the whole thing just crashed. Really sorry about that. I was really just kind of doing the wrap up, and we were just going to do a little bit of question time. So this is going to be really short. This is only going to be like 5 or 10 minutes because we were just wrapping up. So yeah, again, sorry for the interruption. Wasn't the plan. This part of the stream probably won't end up staying up. The other half will, because that contained most of the like meat and potatoes of what we were actually doing. So again, sorry for the weird cutoff. Not sure what happened. If you haven't already seen the stream that we were just doing, I would highly recommend checking it out. We did some really cool stuff. In fact, I'll briefly show that again, just for anyone who's just happening to tune in right now. Yeah, that was interesting. Not really sure what happened. So I'm not sure how many people are actually going to tune into this part because it's really just sort of the wrap up. But if you were just watching the last stream and you had any questions or comments about the thing that we were working on, now is the time. We're gonna do just a few minutes of question time, and then I'm gonna probably wrap it up for the day because we've been at this. We're going on almost 3 hours. So, yeah, that'll be fun. So just for people who are just tuning in or who just don't know what we were doing, this is the thing that we were working on. This is what we just made. The idea is that if you were to take this piece of DNA and put it into a turtle, it would make the outer layer of the turtle turn to glass. And the thing is lagging again. What the hell? Is it working? Or is it just lagging on that end? Weird. No idea. Okay, so yeah, let's do some question time because that sounds fun. Now, yeah, everything seems to be good. It was just like lagging on one of the laptops. Anyway, so we got some questions. Let's answer some questions and then we'll call it for the day. Okay, so we got so it says at the beginning of the stream you said you found plants more interesting. I also prefer working with plants over animals. See, now that is a clear distinction. I do not prefer working with plants over animals. I prefer working with animals over plants. I love mammalian work. I've worked in a mammalian lab before, and I absolutely love it. It is so much fun. I have enjoyed working with plants because they're just so damn weird. And you know they don't have like, and then you broke a gene and then it got cancer, like you do with like mammalian cells. With a plant, it's like, and then you broke a gene, and it didn't care because it had 400 copies of its genome. Or, I mean, I'm hyperbole, obviously. But yeah, plants are weird and robust and interesting. Also, I've got a friend who made like a weird mutant plant thing. And it is just so pretty and so weird looking. How long would it take for the turtle to turn to glass? I have no idea. It would totally depend on what it eats. What type of glass? Pure silica. So like bioglass. How hard is it to start? Is there a way of a community help for this kind of thing? It's not that hard to start. You just pick a project and you start. And it's going to be extremely daunting. And it's going to be very hard. And you're going to have to do an enormous amount of reading, but it's the only way to get going. It's literally just like, this is the whole point of this stream, really. Like, people are like, why are you doing things that you can't test? Just to show the process so that people can pick something ridiculous, like, say, I don't know. Wow, that's weirdly topical. Grow a lens, and then you just, start thinking through it. You go like, okay, what papers can I find to do this thing? They give me this sort of information, like what proteins are involved, what organism do I want to work with, and you just kind of like work through the steps. And there's Facebook groups, there's websites, there's all kinds of stuff to, that people hang out who do this sort of thing. Like if you look for any of the like genetic engineering or like biohacking or DIY bio groups, like you'll find loads of people who do this. Or you can go to like a local community. Well, I mean, now you can't. But when there's not a pandemic going around, you can go to one of the like local community bio labs, and it'll be full of people who do this sort of thing and can help you out. No, not eat sand. There are soluble forms of silica, in usually like silicic acid, that is in food. Many things carry it. Like it's how you get like silica. There's not many soluble silica compounds, but they do exist. What else we got? Will you ever get the DNA that you were designing and test it out? Yes, absolutely. There are certain, certain projects I will end up doing. In fact, the very first stream that we did, that we designed plasmid to do milk and eggs, those ones, the quote is going in today, to basically see it, if we can get it made. And like we did a fundraiser for it, and so we're gonna get those plasmids made and we're gonna actually make them. This silica turtle thing is so ridiculous that it's purely an academic exercise. Thank you, Niles. Terminators and stop codons are dealing with different enzymes. So a stop codon is dealing with the ribosome, so the thing that actually does the turn RNA into protein, whereas a terminator deals with RNA polymerase, which turns DNA into RNA. So they're two separate things that do similar but different jobs. Going back to the beta-carotene yeast, could you use glowing yeast to make glowing bread? No idea. Probably. Shrug. It's the best I got. I don't know. When you say glowing, I assume you mean with like GFP or something. And I've got GFP yeast sitting in my fridge, so I could try this. Is it safe for a teenager to get a magnet implant? Probably not. It's the same thing I said in the video. If you're a teenager, don't do it. Very simple. Can yeast live inside your blood briefly and then you die? Wouldn't the fluorescent proteins break down? It depends which fluorescent protein you're using. If you're using the one from the Odin, then probably. But if you're using like superfolder GFP, which is ridiculously stable, there's a chance that it could survive the process. Or even if it does get denatured, it might renature itself as soon as it cools down, because it's cool like that. What channel did you say the bread video is going up on? The bread video is going on my second channel called The Taste Emporium. There's a link to it on my channel page. I'd highly recommend checking that channel out and subscribing, because it's going to be fantastic. I am super thrilled with how this video is turning out. So yeah, that's gonna be pretty cool. What kind of labs do you want to do with the capture card? I'm thinking like doing some like transformations and like doing like a lot of this stuff where I'm talking about like, oh, you could do a digestion where you just cut with this enzyme. Like I could actually show that live, and eventually it's all going to be into like proper videos. But I think it'd be interesting to do this as I'm working, so like people can ask questions if there's something that's unclear. Where's the Spider Silk project at? I talked about that at length at the beginning of the first part of this stream, which you're going to, unfortunately, you can't just, unwind the clock 10 minutes. You're going to have to actually go to the other stream. But the Spider Silk project is making progress and is very, 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 very frustrating. Yeah, I don't know. I managed to, in theory, get some of the silk DNA into a species of yeast, but it's the wrong species of yeast, so I've got to rebuild part of the DNA to make it work in that species of yeast. and I've got to do a bunch of other mods to it. And it's a whole thing. I'm hoping to have an update video by next month. The only reason I haven't made an update video at this point is simply because I refuse to make that video until I've actually made some goddamn silk. Is basically it. Like, that's the point where I'm at. I'm like, I don't want to do like 4 or 5 more parts where we talk about it failing over and over again. Like, I just want to show it working. Thank you, Max. Greatly appreciated. Okay, I'm gonna do one more because this has been an exhausting process and I'm hungry. So, I got a question. What got you into genetics? And I've said this before. Nausicaa in the Valley of the Wind. It's a fantastic movie. I made it halfway through that movie, and then I signed up to go do bio because I saw it and I was like, I must. I was always interested in genetics. I was like, I don't know. I've always been of the opinion that like I'm made of meat. I should probably learn how to like repair and or modify it. In the same way that like if you bought a car, it would be irresponsible to not know how to fix your car. But also genetics is just wild and wonderful, and I love it. I got some dickhead being like, "Don't be anti-capitalist, okay?" I'm not. I don't like capitalism. I don't. Look at all the wonderful things capitalism has done to the planet. Oh right, it wrecked it. Anything on the neurons? I have said this in every single stream, and I'll probably end up having to say it in every single stream moving forward, but the second part of the neuron video can be found on Ben's channel on Applied Science, and there will be an update when there's an update, which is also going to require the pandemic to kindly leave, which could mean that there might not be an update till 2022, depending on how this continues. So, yeah, that'll be a while. What advice would you give to anyone just starting to learn bio? Try not to get overwhelmed. You're getting muted. Bye-bye. Anyway, if you're just learning bio, try not to get overwhelmed. There's a lot, obviously. And really, I mean, the thing that I can suggest the most is just pick a project and just start working through it. You'll learn way more than trying to like read through a textbook, because you're going to end up having to read through multiple textbooks to get through the yeah, like there's always a lot to learn. We've got another question. Why waste your time pissing about making bespoke things for YouTube streams and not actively contributing to COVID-19 research? Because as much COVID-19 research as is needed is already being done. There's nothing I can contribute meaningfully without millions and millions of dollars. I'm going to be making a COVID video. Chill. Radio telescope update. At some point, I mean, again, the world is on fire and I can't leave my house. So, when that's not a thing. And also so I need it to A, be summer, and B, not be locked in my house. And then I can do radio telescope stuff. Are there any projects that you would recommend for beginners? I mean, there's lots of kits on like AminoBio that are really good. Like I really like the smell one, where you can make like E. coli smell like bananas. I find that one really interesting, and it's because it's just, I find it more interesting than the GFP thing, because you're actually like doing a chemical process rather than just like, ooh, it glows. Not to say the glowing isn't fun, but yeah. So like start with a kit, work through it, get used to doing the process, and then once you're like comfortable using kits, then you can actually like design your own thing and then work from that. The plastic-eating fungus hasn't been touched in literally years. Why is there so much genetic engineering in bacteria compared to archaea? I think the easiest answer is archaea are weird. A lot of them have really weird growing conditions that they like, and there's just not as many. Like, also, it's one of those, like, people figured out E. coli first because that's just what happened. People happen to be growing. It's not actually a great organism. So, I've got a friend who's trying to make plasmids for archaea because it grows in ridiculous amounts of salt. So basically you can use, like, you never need antibiotics in your media. You can just use, like, or like, you don't need to sterilize your media and in media either because it's so salty that nothing else can survive. So, yeah, I mean, I'd love some archaea plasmids. That sounds great. In your last stream, you said you could make a gene gun for cheap. What makes professional versions so expensive? It's the guarantee of function. There's lots of tools you can DIY for way cheaper than a professional one, but a professional one comes with a warranty and a guarantee that it functions. So if you're doing something in like a professional lab where you've got like a grant paying for things, like, you absolutely buy the gene gun. Like, you don't make your own because it's covered. And so you'd rather have a thing that will work and that won't be a source of error in your thing. So it's one less thing you got to chase down. That's really what you're paying for. Okay, I'm doing one or two more and then I'm done. What is the easiest to modify eukaryote? Most mammalian cells, honestly. Modifying them is super easy. You literally, like, mix the DNA with some lipofectamine, throw it into the dish, wait an hour, change the media. You're done. What field of bio do you specialize in? I don't. My whole thing is I don't specialize. Specialization is for insects. There's a reason I do physics and chem and bio and all these other things. I'd rather know lots of things about many different things than know lots of things about one specific thing. It's more useful. What is your favorite form of life? I mean, I like fungus. Fungus are real weird. Like, they're just so weird. But I don't know. I don't really have a specific favorite. I mean, siphonophores are beautiful and weird and wonderful. Lichen are super weird. Because they're like 3 different organisms that are like megazorded into one, which is pretty cool. Can we have a t-shirt for the specialization thing? It's not my line. I mean, sure, but like, it's not my line. I forget who said it first, but there's like a really long, like, it's a much longer quote of like, you should, like, like an adult should be able to like set a bone and do just like this huge long list of things, and then it ends with specialization is for insects. And I would get that as a tattoo. Like, it's my whole existence. Like, I'm only able to do the things that I'm able to do because I have learned about so many different things. It's like when you specialize, you end up hyper focusing and missing solutions to problems constantly. This was the thing that drove me crazy in university. Like, you've got a professor who's been studying the same process for like 30 years and that's all they know. So every time there's a problem, that's the only thing they try throwing at it. It's like if all you have is a hammer, everything looks like a nail. It's that, but with like 30 years of like muscle memory built in. And so you end up missing like huge chunks of useful solutions. I am working on some t-shirt designs. I'm seeing a weirdly high number of requests for a Dangly Bits t-shirt, so I might actually make that just because people seem to be into it. Any Gecko Tape update? No, I haven't touched that project since that video came out, nor do I have any immediate intention to. There's lots of things that I'd love to do. I'd love to come back to the holograms, which I just didn't. The only reason I didn't is because then I need to get an optical table, which is expensive, and I literally did not have a location which was stable enough to comfortably be willing to do that. However, because of the pandemic, where there's all of a sudden way less car traffic, I might actually be able to make it happen, but there's more people in the building, so them walking around could mess with it. Holograms are a pain in the ass, which is why I haven't come back to that. Gecko Tape is in a similar realm. Any improvements beyond what I did would be getting into, like, circumstances would need to be different. Okay, let's do one more, and then I'm done. Let's see what we got. Yeah, it's Heinlein. Thank you, comment people. Was it Robert Heinlein? Yeah, Robert Heinlein was the person who said that. Is it true that bananas looked ugly until they were genetically modified? So bananas aren't genetically modified. They're just weird. The bananas that you eat, you will notice, are distinctly seedless. Those little tiny black specks in the middle are the remnants of the seeds. So they don't produce seeds. They're multiplied through cloning. So every banana you've ever eaten is genetically identical, unless you've eaten a different species. Whereas originally the Gros Michel, which is the old kind of banana, which we don't have anymore because it died to blight, actually had seeds. So it wasn't that they were ugly. All bananas basically looked the same. Just the Cavendish was a good size and tasted reasonably and was easy enough to transport is the only reason we're using that one. Also, yeah, it was bred. But I mean, it's a cross between two different kinds of bananas that ended up with a sterile clone with the properties that we wanted. So it has to just be it has to be propagated asexually. Like, you can't breed it anymore, which is also why we may end up losing it, because there's currently a blight going around that kills bananas. And because they're all genetically identical, it really kills the shit out of the bananas. So if you discover that disease, you basically torch the entire field, because it's the only way to deal with it. Okay, well, that was our last one. Yeah, in some countries, bananas still have seeds. Lots of varieties of bananas still have seeds. Just the mass market Cavendish banana does not, and is the most common. Okay, anyways, that was our last one. I did not give up on the spider thing. I am literally actively working on the spider thing. It's like, if I wasn't editing, I would be working on it today. I've definitely not been. I've been dreaming of the spider silk thing for 6 years. I'm not going to give up because I got stuck. I'm making progress. I've talked about this at length in other streams. But anyways, I'm going to wrap it up because this has been a long stream. We've been going for 3 hours. We made a really cool thing. It'll be up on GitHub. If you've got suggestions for future topics, leave them on Twitter. Use the hashtag. Just send them to me on Twitter. Use the hashtag Who'sGene, and they will get added. They will get added to the hat, and then potentially get pulled on the next stream. The next stream will be on Friday at 2 p.m. Eastern, same as always, and I think it'll be a lot of fun. I hope you guys are staying safe. Try not to go too crazy. Stay safe. Stay happy. Do some cool bio stuff if you can. If not, maybe pick up a nice bio book. And yeah, that's where I'm gonna leave it for today. I hope you guys all enjoyed. If you missed the earlier part of the stream, be sure to go check that out. Be sure to go over to my second channel, The Taste Emporium, and subscribe. There'll be a new video out tomorrow. And yeah, that's it. I'll see you guys next time. Have a great day.